सेव किया था ना?
मैं सिर्फ इसलिए गया था क्योंकि उसने मुझे कॉल किया, that's it. But listen तो.
Save it for someone else.
जिंदगी में सेव करना जरूरी है, मगर जब बात पैसों की हो, सिर्फ सेविंग से काम नहीं चलेगा। इक्विटी म्यूचुअल फंड्स में इन्वेस्ट कीजिए और अपने सेविंग्स को आगे बढ़ने का मौका दीजिए।
Mutual fund investments are subject to market risks. Read all scheme-related documents carefully.
It's tough for me to figure out how interested you are in sciences or biosciences and further on botanical sciences. I know that all of us had got obsessed with virology at one point. All of us had become amateur virologists when COVID was raging. But plant botany, who cares, right? But you know what? Almost all of our food comes from plants. Even if we are meat eaters, even the animals whose meat is in the food chain also eat plants. So plants are very important. So crops, our wheat, our rice, our vegetables. Also equally importantly, edible oil. Edible oil is very important for us. And India, you know what? India is very short of edible oil. India produces just about 8 million tons of edible oil and imports almost 15 million tons, which means India produces about 1/3 of its edible oil, 2/3 is imported. Last financial year, that import bill was about $19 billion. So India has a trade deficit not just on the import of one kind of oil that's more familiar to us, the geopolitical oil, that is the crude oil, but India also has a big deficit on edible oils. Now, what is the solution? So one solution we've talked about is a very interesting government scheme to set up new areas in India where palm oil can be grown. But what about oils which grow in India traditionally? Can we grow more of those? One of those is mustard oil. Mustard oil is a popular oil in large parts of the country. It's a very interesting frying oil also for pakoras and stuff like that. But India has a short supply of mustard oil. India can also produce more mustard oil. But once again, Indian agriculture has had a problem for almost 45-50 years now. That is after the peak of the Green Revolution. When Green Revolution peaked, India's yields and productivity on our farms has remained about the same. It has stagnated because Green Revolution had given us such surpluses that we generally thought, ab kaam ho gaya, no need for any more, population is increasing, but India is producing enough wheat and rice and other stuff, so why bother? But in the process, India fell short of many things. The biggest problem that India, the biggest setback or the biggest gap that India created all this while was productivity. And one of the reasons was that Indian science fell behind. So the same science which has given us the great hybrids, dwarf wheat, dwarf rice, etc., which revolutionized the production of those grains and which made sure that India did not have mass starvation. Remember, China has had mass starvation and huge famines. Why? They were short of grain in the 60s and late 50s, as India was short of grain in the late 50s and 60s, but India imported its grain from the US and elsewhere. China did not import. So they starved. India avoided that catastrophe going ahead. Now, can India do, say, similar revolution with edible oil? So one group of scientists in Delhi, in a lab of Delhi University, run a center called Center for Genetic Manipulation of Crop Plants, CGMCP. And there, Dr. Deepak Pental, who's a former Vice Chancellor of Delhi University, he has been working with a group of very fine scientists, who I call sometimes the famous five of Indian genetic science plus five. So five scientists and Dr. Pental himself. They've been working for almost 30 years. Team members have come and gone for almost 30 years on genetically modifying Indian mustard and genetically modifying Indian mustard in a way that its yield goes up. It also becomes resistant to some common diseases, particularly a stem borer and also a kind of a rust. Rust is usually a fungus. So they've been working on this project. At the same time, there's been such fear of GM foods, GM seeds. India still has not approved any GM food. The only genetically modified seed that India has approved in India is cotton. So while we know that we don't eat cotton, but it's also a misnomer to say that is not part of the food chain, because cotton seed is then essential to our cattle feed, and we drink the milk of those cattle. And so those cattle, by way of their milk, ghee, paneer, dahi, etc., are part of the food chain. But also because cotton seed oil also goes into our edible oil mixes. So now what these scientists have done is they produced a genetically modified, or as Dr. Pental prefers to say, genetically engineered mustard seed, which is called DMH11. That DMH11, the work has gone on for almost 30 years. This lab has been working as one unit for 20 years. Dr. Pental, even after he ceased to be Vice Chancellor, he could have taken some sinecure in UGC or something, etc. But he is a scientist from his heart. So he continued on working on his lab. So they produced this seed, and so far, because of a lot of activism and activist opposition, and also opposition from Swadeshi Jagran Manch. So this was a strange axis, if I may use that word, of Luddites on the left and the right. So Luddites were people 200 years ago in Britain who used to fear engineering, new technology, new stuff that was coming in as a result of the Industrial Revolution. So that word has stuck. So Luddites of the left and right unite. We have nothing to lose but our common sense. And that's what happened, and that's why this technology had been held back. Modi government now, all credit to it, after hesitating for a long time, has now approved the planting of these seeds out on farms. It is still not been made commercially available, but what's happened now is, with this permission, these seeds can be planted in open fields and their studies can be carried out. Finally, these seeds have got final safety clearance from Genetic Engineering Approval Committee. That's a very high level committee of Government of India, GEAC, in 2016. In fact, it was in 2016, after this approval, that I recorded a walk the talk with Dr. Pental and his team at their lab, and I will share a link with you so you can watch that episode of Walk the Talk. But it's taken Modi government 6 years after that now to approve it for planting in the farms. Now, planting in the farms, these new tests or these new safety checks, because you have to be very careful, there is a lot of suspicion about genetically modified crops. They will now see what is the effect of these plants being grown in the open on the environment, on insect life, particularly honeybee life. Because honeybees suck a lot of sap from mustard plants. That's why wherever you have large mustard crops, large expanses of mustard crops, you also have farmers producing mustard honey, because honeybees sit on mustard flowers and that honey also has the flavor of mustard. So there's been a fear, particularly among activists, that if you bring in these genetically modified crops, then honeybees that sit on them will lose fertility or something awful will happen to them, or maybe honeybees will become some butterflies who will then grow into some giant monsters or maybe vultures or something like that. So there are all these fears. So these tests now, field trials now, are supposed to set those aside. Now, while this goes on, we need to understand first of all the science of this. And when I explain the science of this, you might think a little bit that I'm taking you back to the days and the months when we used to talk about new researches every day, new findings on coronavirus and also new vaccines, on immunity, immunotherapy, etc., etc. So it's a bit like that, because science looks complex, but science can also be very simple and quite fascinating. So what do we have here? We have a popular Indian mustard variety called Varuna. Varuna is among the more popular, higher yielding Indian mustard varieties. How can you make it even higher yielding? Now, usually scientists now for many years, in fact, the turn of the century, those who've studied basic science in their school would know Mendel's law, would know when you cross two plants, one set of offsprings comes out taking some characters from one, some from the other. That is called F1, or first filial hybrid. And usually you find when you cross plants of maybe the same species but which are genetically different. For example, you can cross one kind of wheat with another kind of wheat. you can sometimes produce hybrids which have greater vigor, which are stronger. That is called hybrid vigor. So why couldn't you do this with mustard? If you can do it with wheat and so many other crops, why couldn't you do it with mustard? You couldn't do it with mustard because mustard has a peculiar issue. If you want to crossbreed plants, you first should be able to breed a plant. Breed a plant means you should be able to pollinate one plant with the other, in which case you have the female reproductive organs of one plant exposed to the sperm from the other. That's how you carry out cross pollination. And then if you take another variety there, I'm oversimplifying it, then you can carry out hybridization. So instead of taking two wheat plants of exactly the same type, you can take one of one type, one of the other, you can produce a hybrid. What you should be able to pollinate one with the other, cross one with the other, or for simplicity, marry one with the other. Or okay, in these times maybe marriage is not the most perfect way to describe it, at least impregnate one with the other. The challenge with the mustard flower is that mustard flower is what botany textbooks would describe as a hermaphrodite. Hermaphrodite means the mustard flower has both the female and the male organs within it in the same flower. So pistil, which is the female organ, see the pictures, and stamen, which is the male part, are both in the same flower. So as the pistil opens up to receive the sperm or the pollen is sitting right there and falls there. So there is no time, no possibility for the bees. Bees are the most popular pollinators of plants. So even the bees who go from plant to plant, even they find it tough to bring in pollen from one plant because pollen gets stuck to the bees' feet and mouth parts, etc. Even they are not able to cross-pollinate one plant with the other because the plant is self-contained. Male and female are both in one. To cross-pollinate it, either with the same type, which won't make so much sense, or with another type, which might create a hybrid, which gives you hybrid vigor, in which case higher yield and more disease resistance. You have to do something to this plant, say Varuna in this case, so it doesn't pollinate itself. So it doesn't feel so satisfied that I am male, I am female, life is fun, where is the problem? So for that, so the scientists had to find a way of sort of interrupting this self-indulgent fun. I am using very easy language to simplify complex science to you. How did they do it? They found a bunch of genes. In fact, they found two genes. They found two genes in a bacterium which is found in our soil, right? Commonly found bacterium in the soil called Bacillus amyloliquefaciens. Bacillus amyloliquefaciens. You can see the full name on my screen. Now, Bacillus amyloliquefaciens, scientists have found for a long time in any cell, it has a gene. That gene is called Barnase. That gene is a protein. That gene is like a natural antibiotic protein. So Barnase eats up RNA, right? It's like kills a bug, kills a germ, kills a bacteria, kills a cell. Now the scientists also find that while Barnase eats up RNA, it's not able to do so in its own cell. Because if the Barnase gene is sitting in the cell and it's eating up the RNA, because it's oozing out of the ribonucleus, I will not go into more detail because that will get too complex. But basically think about it. This is sitting in a cell. A cell is a living being. It's oozing inside the cell. It can eat up the RNA. If it eats up the RNA, the cell will die. So how come the cell has this time bomb inside it which keeps blowing up all the time and yet the cell survives? So cell must have something to counter this. That something was another gene called bar star. That element was another gene which the scientists name bar star. Now bar star remains tied to Barnase, and bar star basically is the antidote to Barnase. Bar star does not allow Barnase to play the antibiotic within its own cell, within its own home, and play havoc with itself. Otherwise, Barnase will become the equivalent of, like, you might say, a human bomb. It blows up and everything else blows up around it. But bar star keeps this sort of human bomb equivalent tied down. So these scientists and Dr. Pental's team took out these two genes, that is Barnase and bar star. Barnase, the destroyer, bar star, the blocker. That is the same bacillus, Bacillus amyloliquefaciens. Now what did they do? So they took out Barnase, which is the destroyer, and put Barnase in a cell of the Varuna variety, the Indian variety. That cell was then cultured into a plant. It became a plant. The plant had this gene. Because the plant had this gene and Barnase is a destroyer, in this plant then Barnase went into destroying the sperm or deactivating the sperm in the stamen of the flowers of this plant. So this plant then could not pollinate itself. So that comfort that I'm married to myself, I can produce my own babies, that comfort was taken away. I know I'm oversimplifying it, but that is the best way I think to understand this. Now, how does this plant get pregnant? Because this plant is now purely female. So you then take sperm from another plant, the other variety with which you want to hybridize this Varuna. And that other variety was again a very familiar East European variety called early Hira. Some of these are called by other names also, but basically East European varieties which had other characteristics. And in the lab it was seen that this cross fertilization or this hybridization gave you what is called as hybrid vigor. These seeds produced plants which gave you more oil, which were also hardier when faced with other disease, funguses, bacteria, etc. So you might ask me, if the scientists then took out sperm or pollen from this East European variety and impregnated the Indian variety, why would the same Barnase gene which was already in the Indian variety also not destroy the sperm coming in from the East European variety? Because scientists are smart. As we told you earlier, remember, scientists have taken out not one but two genes from that bacillus. One gene was the destroyer, again other gene was the blocker or the antidote. So that gene has been inserted into this East European variety. So East European variety, when you use sperm or pollen from this plant to impregnate or pollinate the other, so then this pollen goes into the female plant, purely female plant, and basically says, look, where is Barnase? You want to stop me, but I've got bar star, right? I will stop you because bar star stops all action of Barnase. So one takes away the other's power. So Barnase was first used to render this one variety purely female, and bar star is now used once again to make sure that this female pistil is able to receive this pollen and get impregnated and can produce seeds. These seeds then carry the new characteristics. So these seeds can give you new crops with higher yield. So that is exactly what has happened right now. So once again, I know it can get too complicated, so I'm walking this gingerly. So as these two genes are inserted in these plants, another gene comes in as a kind of excess baggage, that is called bar genes. So three genes coming. Now bar genes has a special characteristic. What bar genes does, first of all, it helps you identify which plant has been genetically modified. So genetically modified plants don't get mixed up with others. So bar has to be added to make sure, even by scientists, that they are conducting experiments on genetically modified plants, because all mustard plants otherwise look the same. But the bar gene also has another characteristic. It can make these plants hardy or resistant to popular weedicides. Now activists have a lot of concern about weedicides. Now weedicides in India are used, say, in rice crops. But activists worry about weedicides because they think that if you use genetic modification to resist weedicides or make weedicide proof plants, and God knows what else can this be misused for. So these ones then are not permitted, these plants with the bar gene are not permitted to be commercially cultivated. These are only for scientific explanation. Now, I had a long conversation, I've had many long conversations with Dr. Pental and other scientists on how unsafe can these things be, how much should we worry about them. So first of all, Dr. Pental, You will see this still from that walk the talk I recorded with him. The presentation they made to Genetic Engineering Approval Committee was 4,000 pages, and a lot of detail was sought and a lot of detail was given. Second, he said that since 1996, trillions and trillions of meals of genetically modified foods. See, the US, Canada, lots of Africa now, Southern America, Latin America, a lot of countries now have large amounts of genetically produced food, particularly soybean, for example. You will soon see a detailed story from my colleague Sayantan Bera. He's just been to these big soybean farms in America, and he's written about them. That soybean farmer in America gets 3.5 times the yield of the Indian farmer. So Indian farmers simply can't compete. But that apart, if you just look at the science, Dr. Pental now tells me that now we have enough testing available in science to check any protein for its allergenicity and toxicity. And he says that to look, 99.999% proteins are neither toxic nor allergenic. So you don't need to worry about them. And once again, you can test them scientifically in a lab to 1/1,000,000th of molecular dilution, which means 1/1,000,000th of part of a molecule also you can test and figure out if it's dangerous or not. Second, while we've been caught up in this activism and doubts and Ludditism, the Chinese have gone way ahead of us. The Chinese are now almost a generation ahead of India on seed and scientific research. Remember something that I've said a couple of times? Chris Wray, the head of FBI, he has said more than once that the Chinese have sent their spies deep into the American countryside, Midwest, and again the large agricultural areas, who've actually dug the soil to pull out genetically modified seeds, so they can then take it back, reverse engineer it, and save billions and billions of dollars of research. And by the way, do you know how much has this lab cost, Government of India? The lab which produced this mustard, it's just about 50 to 60 crores, right? So for just about $7 billion, you produce something that costs billions in international research. We in India have been caught up in this activism of driving Monsanto out, and we've succeeded in driving Monsanto out. But you know what? Monsanto has been acquired by Bayer, the big multinational company, for how much? $63 billion. That's how much this research is worth. At the same time, while we've been driving out Monsanto and blocking so much of seed and biotechnology and plant genetics research, the Chinese have acquired one of the biggest biogenetics seed research firms in the world. That is Syngenta. Syngenta is now a Chinese company. How much did the Chinese pay for it? They paid $43 billion for it. In this environment, you have these scientists on their modest, humble government salaries, who produced this marvelous research for you for just about $7 billion. And India has several of those agricultural scientists. Unfortunately, this field has not grown as much as it should have after the Green Revolution because we got lazy. But there is also Dr. A.K. Singh, who is director of Indian Agricultural Research Institute in Delhi, what is called also Pusa Institute. He and his team have also developed these wonderful direct seeding basmati varieties. And this year we will see the first big crops of these, because these are direct seeding basmati varieties. You will see some pictures on my screen. Direct seeding basmati varieties means that you don't have to grow a nursery and then transplant them. Because when you grow a nursery and you have to transplant, then you have to flood that nursery. You use a lot of water to prevent weeds from growing. But if you do direct seeding, then you don't have to worry about that. And that's when the use of water, you will still use a lot of water for rice, but the use of water becomes less. So that development we were already celebrating, and now there is something else, which is India's first genetically modified crop research or seed research, finding government's approval, government's clearance to move towards commercialization.